Bandyopadhyay sir for accepting our invitation to deliver the 24th lecture of Indian Phytopathological Society Platinum Jubilee Lecture Series. Now I request Dr. Sushil Kumar Sarma to give welcome address and the introduction of the speaker.
Sushil, your voice is not coming. Sushil. No.
Sushil?
You check your speakers.
If not, then it's fine. I will take it up. Sir, I'm Dr. Prashant Zambulkar, Associate Professor Plant Pathology, and I, on behalf of Indian Phytopathological Society, I welcome you on this Platinum Lecture Jubilee series, and it's a 24 lecture. It's our great privilege that you have accepted our invitation to deliver lecture. Now I take this opportunity to introduce you as a speaker for this lecture. You are a principal plant pathologist at ITA and Ibdan, Nigeria. You did your BSc honors and agriculture in animal husbandry from Jubilee Agriculture University Technology of Pantnagar, then MSc in plant pathology from Jubilee University of Agriculture and Technology in 1976, and PhD from Hisar, Haryana Agriculture University in 1980. You have held so many positions, right from as a scientist in ICRISAT to principal plant pathologist at ITA, Ibnan, Nigeria. That's a great thing. And you have been awarded for your excellent contribution to the science of agriculture with so many awards, including right from the most coveted award for the students like that. It starts our, we know as a plant pathologist, our journey starts with this award. If we are fortunate enough to get that MJ Narasimhan award in plant from IPS. So I told you, usually I tell to my students also, if you are a plant pathology student, that your journey start as a plant pathologist with this award. Right. So it's nice. So then you have been awarded with outstanding scientist, means thrice in 2010, 2011, and you have been awarded with Excellence in International Service Award by American Phytopathological Society. So nice of you, sir. So you have been a fellow of American Phytopathological Society too, and you are working on development and commercialization of biological control of aflatoxin, integrated mycotoxin management, in African agriculture, disease of tropical crops like sorghum, banana, yam, cassava, and so many. And you have experience of working on breeding of disease-resistant varieties also. So it will be a great opportunity for all of us to hear you, sir. I'll request you to share your slides and please start your presentation. Well, you're most welcome, sir. Thank you.
Thank you very much. Now you're right. The first award that I got was the Narasimha Award. That's the most, and I cherish that, and I have that on my wall actually. So it was a great honor to actually receive that as for my student, for my PhD research. What I'm going to speak today is about 20 years of our research on biocontrol-centric aflatoxin mitigation in Africa. I've got a buzzword here, more than 1 million reached. I'll tell you what this actually means later on. This is the work of a team, and I'll show you pictures of the team later on in the seminar itself. And without the team, this work wouldn't have been possible. I mean, both within IITA and within various national and international partners. Now, I have to figure out how to advance the screen. Right. So the picture that you see here is a Kenyan woman, actually a grandmother. And this was one of the first visits that I did in Kenya in 2004, if I'm not mistaken. And I met this lady who was standing in front of a pile of maize bags. She had tragedy in her life. And Lord. I have to, yeah. Right. So, I'm sorry, the floor went off. So she had the tragedy in her life. Just about 2 years ago, there were about 125 Kenyans, they actually died of aflatoxin poisoning. And our family members were among them. And the family member, her son, actually left their grandsons and their granddaughters, and she was responsible for them. And the way she actually handled their school fee was to sell maize in the market, but she couldn't sell this maize in the market because about 3.5 million bags of maize was declared as contaminated. And she was sitting on it and she didn't know where to sell it. The only option was for her was to actually either feed it to animals or consume it themselves. And so aflatoxin is not something abstract, it actually hurts people's lives. Now, the impact of aflatoxin, give me a moment, I'm going to change my screen from, just give me a moment because this slide sharing is not working. Oh, come on. Is the screen visible now?
Yes, sir, visible, sir. Come to slide mode, sir.
Yeah, no, I'll do that.
Yeah, now visible, sir.
Yeah. So aflatoxin actually in Africa, it cuts across 3 different sectors. It impacts on public health, It impacts on trade and economy and on food and nutrition security. Nearly 30% of all liver cancer cases in Africa can be attributed to aflatoxins. About 40% of all commodities in the local African markets sometimes exceed the allowable levels in foods. And Africa loses, on a conservative note, about $100 million annually from lost potential export. And so it does actually have an impact. I'll show you later on in more details of what these impacts are. And as we all know, that the aflatoxin is actually produced by, it's a toxic chemical produced by various species of Aspergillus. It causes liver cancer, stunting, immune suppression, death, as I mentioned earlier. It curtails animal productivity, and it's regulated in the market at very low levels, in parts per billion level. It actually grabs a lot of headlines in Africa. For example, food poisoning, the last news item that you see, about 14 people died in Tanzania a few years ago. And oftentimes the agro-processing factories, they reject maize produced locally, and they have to import the maize to meet the quality requirements of their products. In worst cases, the regulators sometimes blacklist companies to reduce aflatoxin in the market. And very recently, there were tensions among countries in East Africa when Kenya banned maize import from Uganda, Tanzania, and it became quite a huge political issue. So it does actually impact on various facets of life and agriculture and health and trade and economy. These are some examples of deaths that have happened over the last many years, including in India. But as you can see, most of the deaths have actually taken place in Africa, primarily in Uganda, Tanzania, and most often in Kenya. Aflatoxin is very common in the African food system, and aflatoxin consumption would oftentimes lead to exposure of the body to aflatoxin. And what you can see here is the proportion of children in Benin and Togo. 99%, 98%, 100% children are exposed to aflatoxin. And the exposure is at very high levels too. Similarly, in Tanzania, the same kind of effect was found, but not as much as in West Africa. There have been studies, many studies more recently actually, that showed the association between exposure to aflatoxin and immune suppression to low birth weight, stillbirth and preterm babies, reduced weight and height among the babies, and low growth rates in several countries. And so this is more or less known right now. In addition to that, a large number of cases, as I mentioned, 30% of the cases can be attributed to aflatoxins in Africa. In Nigeria itself, about 8,000 cases of liver cancer are noticed, and it costs in between $380 million and $3.1 billion to the national economy. Similarly, in Tanzania. It hurts the animal industries, as I mentioned, and they are the most aware among the various sectors in agriculture about the harmful effects of aflatoxin, because they can see the chicken die because of aflatoxin exposure. If you look at the trade, these two graphs actually show the decrease in trade due to aflatoxin and other reasons too, in Nigeria and in Senegal. In Nigeria, about 99.6% decrease in export was noticed from 1970 to, I think that the figure here is 2018. And in case of groundnut, reduction is about 48%. In the 70s, one could see these pyramids, what they called as groundnut pyramids in northern Nigeria. You don't see them anymore because these railroads used to actually carry the groundnut bags to Europe. After the Europeans enacted aflatoxin regulation at 4 parts per billion, Nigeria just couldn't, and many of the African countries just couldn't meet the standards, and as a result, the export actually dwindled. But this is the effect of aflatoxin. So if one can actually control aflatoxin, for example, this study was actually is from Senegal, where the World Bank estimated that if the country invested $4.1 million in capital investment and 15% recurring cost for aflatoxin control, that would actually translate to a 30% price differential for oil cake. And the export would increase from 25,000 tons to about 210,000 tons. And that would add about $281 million to the exchequer just because of this small amount of capital investment. So it actually does make sense to control aflatoxin in various countries. But the export also comes with a downside. And oftentimes, what is exported is best. And this is what you are seeing as women actually sorting out the good grains from the bad grains. This is in Mozambique and in Senegal. And as a result, the local population are exposed to high levels of aflatoxin. And the best quality grains are exported to Europe, where it is fed to birds. And therefore, we do have a moral and economic reason to address aflatoxins in Africa due to health, trade, food and nutrition security considerations. Now, aflatoxin affects many crops. Maize and groundnut are the best known. Sorghum, cottonseed, and many other crops are actually affected. And in the US, almonds and pistachio, they are affected, and their exports are pretty badly affected because of aflatoxin. This is a picture that my colleague Alejandro took in Senegal, where you see this mountain of groundnut that are ready to be shipped for processing purpose to the factories. As a plant pathologist, we normally dig deeper in, and so we dug in deeper. And this is what we found in these peanuts. This is aflatoxin contaminated peanuts here. And you can imagine what would happen if this pile of peanut is processed into food or now, the Partnership for Aflatoxin Control in Africa, they conducted a 4-year study, and in several countries where they sampled crops at various points, and they found that in these countries, about 72% to 96% of the samples were positive for aflatoxin. And if you groundnut, in many countries, the limit is 15 parts per billion. About 15% to 70% of the samples were beyond the limit, what would be acceptable in market. And on an average, 37% of the samples were beyond the limit. If you take the European Union standard of 4 parts per billion, 61% just couldn't meet the standard. And if you take the EU, the codex limit, 33% just couldn't meet the standard. Similar study was done in case of maize, and similarly high levels of aflatoxin contamination was noticed. About 23% of the samples couldn't meet the codex limit. About half the sample couldn't meet the European Union standard. So it is quite common, the exposure and the contamination is quite common in the African agricultural system. Aflatoxin problem actually starts in the field. And so this is some data that was taken some way back, where we looked at peanut and maize at harvest and after harvest. but the data that is shown here is at harvest. What we found was about 54% of the samples were beyond the limit of the European Union. 70% of the maize samples were beyond the limit of European Union. If you look at the amount of contamination gain, it is pretty steep actually, about 3,400 parts per billion in peanut, about 840 parts per billion in case of maize. And the point is that this is at harvest itself. And this actually increases when it is stored improperly. And so, as you can imagine, the contamination will be quite high in the food systems. There are many ways aflatoxin can be controlled, right from planting up to consumption, and the entire in production chain, including processing. I'll not go into the details, but I will just mention a few in the next few slides. But whatever the control measures are, they must be rooted to awareness, institutions, and policies. All the technologies, they must be supported by institutions and policies. And awareness of the problem, which is very low in the African system, must be actually improved and must be considered to implement aflatoxin management systems. So we are actually designing these strategies that are centered on biocontrol, and this would be the main topic of the presentation. Now, in terms of the grain drying, some interesting developments are actually taking place in that African system, where the mobile dryers are being, people are trying to popularize them, but there are many types of drying, but unfortunately, none of them are actually practical and none of them are being scaled up in Africa. But this is the most common one, drying on tarps, that is practiced in the African households. In terms of storage, there are many kinds of storage. These are the traditional storage systems in Africa, but there are many kinds of storage systems. And the improved storage, like the PICS bags, the hermetically stored bags, the PICS bags, the grain core bags, plus there are plastic containers and metal silos. And these are actually effective compared to the most common one, which is the storage in polypropylene bag. And this is a study conducted by Aflatoxin project, where they found, this is in Kenya actually. So all the devices that I mentioned earlier, they do reduce aflatoxin contamination. But what I would like you to actually see is at the beginning of the storage, the level of toxin and contamination was nearly 2,000 parts per billion. And in Kenya, the safe limit is 10 parts per billion. So by the time the crop was being stored, it was already contaminated. But despite that, our 6 months after storage, the increase in the amount of contamination was very low in the improved storage devices compared to polypropylene bags, where the increase was more than double. So good storage and good drying and good storage does help. And all those practices put together reduce aflatoxin and fibonacin and improve health. And these have been proven. So these technologies are available and these technologies can be used. But the main question is, how do you scale them up? In experimental situations, small scale, research purposes, they work. The main thing is about scaling up. So effective technologies are available and more work is required on drying. Institutions are also important, and there are different kinds, different scale and type of institutions. In terms of scale, we have international, national, regional organizations that are active in promoting aflatoxin control. And there are different types of organizations, like the National Research Organizations, the national organizations, the NGOs, the And what is now encouraging is to see many of the private sector partners, they are coming into the picture and implementing aflatoxin control for the industrial purposes. One of the positive things that had happened in the African is in the institutions, establishment of the Partnership for Aflatoxin Control in Africa at the African Union level. And that actually created a large amount of impetus on aflatoxin control among various countries. What is a little disturbing is that the health institutions are less involved in the aflatoxin control effort because I think they consider other problems to be of higher importance. In terms of policy, there are again various kinds of organizations that are involved in policy making. I mean, Codex is, we all know about Codex that implements the maximum residue levels. FAO has got a large number of codes of practices to control aflatoxin. Food safety laws are being implemented. I mean, India has a good food safety law. We have actually created policy briefs for the East African community, the 6 countries in East Africa that form the East African community. And you can see the series of papers that we wrote for East African community. The regional economic commissions, the West African one, ECOWAS, the Southern and East African one, COMESA, and the East African one, EAC, East African community, they have actually created aflatoxin control plan. And that actually shows the importance that these regional economic commissions put aflatoxin in. Many of the countries have included aflatoxin mitigation in the National Agriculture Food Security Investment Plans, which are the national budgets actually. And so you will see a line item on aflatoxin mitigation in these countries. However, we do have these policies where oftentimes implementations of the policy is the crux of the issue actually. Awareness and training are also important, communication, and many efforts have been done, but I think more is required. I mean, there have been manuals and guidelines, videos, posters, field training, short ads, and so lots of efforts have gone in there. But more is required because awareness creation is not one shot; it's a continuous. It's a continuous effort. Now, let me come to biocontrol now, because I've talked about everything else. Now, the other technology that we work on is biocontrol. Now, in terms of biocontrol, as I mentioned earlier, aflatoxin is produced by Aspergillus, a species of Aspergillus, and most often Aspergillus flavus and Aspergillus and parasiticus. There are many other strains of Aspergillus that are, at least one of them is not characterized as yet. But there are other new ones that have been named that produce toxins. So, but in nature, you have some strains that produce large amount of toxins. These are toxigenic strains, and others don't produce the toxin. These are the atoxigenic strains. So in the graph that you see here, so you have the log concentration of aflatoxin production, and these are each isolate, the 54 isolates. And if you look at it, there's in most of the strains, isolates produce large amount of aflatoxin, but there are a few blanks here. This doesn't mean that they have not been actually analyzed. They were analyzed, but they don't produce any aflatoxin, so they are atoxigenic. These atoxigenic strains are already present in the crop, and if we can somehow increase the frequency of the atoxigenic strains, we can displace the atoxigenic strain and toxigenic strains from the field, we can decrease aflatoxin contamination. For example, in a field, the isolates of the applied strain, biocontrol strain, if they increase, the amount of aflatoxin actually goes down as a proportion. So if we can increase the proportion of atoxigenic strain in the field, we can decrease the amount of aflatoxin. And this is the basic principle of biocontrol. We have shown that atoxigenic strains can be applied without actually increasing the overall quantity of A flavors in the crop or in the environment. So the amount of Aspergillus population remains the same, but the proportion of atoxigenic strains increase vis-a-vis the toxigenic strains. And these strains actually move from field to the store. That means what we apply in the field, the biocontrol is applied in the field before flowering. And if it is applied in the field, the strains get onto the developing grains and it's carried over to the stores after harvest. So the protection continues from the field to the store. Now, I will not go into the details of this, but just to mention that we do have a pretty robust selection system of atoxigenic strains. We start with almost 5,000 strains from each country and look for strains that don't produce aflatoxin. And then we group them genetically into various VCG or SSR groups, and then look for those groups that have got wide geographic distribution and no toxigenic member in them. Then we sequence the aflatoxin production pathway and genes in the pathway, and then look for strains that have got defects in the gene cluster. The other thing is that we do some lab assays to show that they actually outcompete toxigenic strains. And based on the selection criteria, we select about 8 to 12 native strains for field tests. I must actually emphasize that these strains are native strains in the sense that they are found in the same country for which the biocontrol products are being developed. But all these assays are being done in the lab. But biocontrol actually takes place in the field. So we have to now evaluate these strains, look at their field efficacy. And so we then look for among these strains superior capacity to colonize, multiply, and survive in soil, and how often they can be isolated from the grains, and how efficient they are in terms of reducing the aflatoxin contamination. So based on this initial field study, from 8 to 12 strain, we select 4 strains and create what we call as multi-strain product, and we call them AflaSafe. So, but AflaSafe is, each country has its own version of AflaSafe containing their own active ingredients, native active ingredients. Now, this is an example of the product in Nigeria, AflaSafe, and there are 4 strains, or the 4 active ingredients, and they have deletions in the aflatoxin gene cluster, and they have SNPs in the aflatoxin gene cluster. And in some cases, the entire 26 genes are actually missing. Like this particular strain, OG022, the entire gene cluster is actually missing. So there is a genetic basis of why these strains are atoxigenic. Now, in terms of how we produce AflaSafe, we take the 4 native genotypes, we multiply them in sorghum grains in bottles. And so this is the sorghum grain, and after 7 days of multiplication, it turns completely a green mass. We make spore suspension, and into it, the spore suspension, we add a blue color and a sticker. And this suspension is then combined with heat-killed sorghum grain in a seed treater. And then what we get is a product that looks like this, a blue-colored product, which is basically sorghum grain coated with atoxigenic strains of Aspergillus flavus that is ready now for application by farmers. Now, how does actually it work? Again, I'll come back to it. So in a non-treated field, the Aspergillus population, it actually increases after a particular stage. And in these non-treated fields, there is higher proportion of toxin-producing strains compared to non-toxin-producing strains. The treated field, Aflatoxin is applied just before the normal population of Aspergillus actually increases. It is applied at the rate of 10 kg per hectare, 2 to 3 weeks before flowering, and there is only one application done, and the cost is in between $15 to $20 per hectare. And once it is applied to the field, the product, the sorghum, the carrier, it begins to sporulate, and these spores spread out in the field. And after spreading out in the field, most of the population that increases later on are actually safe strains. And as a result, we have very little toxin-producing strains, and more of them are atoxigenic strains, resulting in little or no aflatoxin. But the key thing is that the crop fungal content is more or less the same between the treated and untreated crops. Now, we have generated efficacy data from more than 9,000 fields, but I'm just giving you one of the commercial applications, a study that we did for 4 years. And this study was done with grain lots, and each lot is a 30-ton lot, okay, in various years. So we are talking about a 10,000 tons lot here and almost 100,000 tons of lot here. And the number of analysis are these number of analysis. We did see that in the control, the amount of aflatoxin was higher, but in the treated, the amount of aflatoxin was below 4 parts per billion on an average. But in some fields, of course, you'll have more than 4 parts per billion, 15, 20 parts per billion. But on an average, we have less than 4 parts per billion. And another thing is that the higher the aflatoxin contamination in the field, more effective is the biocontrol. Because you see, with the 53 ppb level of aflatoxin in control, we had a 93% reduction compared to 6.1 and 1.7. And another aspect is that, aflatoxin is highly variable, occurrence is highly variable. So what happens with biocontrol is we actually create a safe environment in terms of the fungal population. And as a result, the variance in between samples actually goes down pretty dramatically. And as you can see, in between treated and control, the mean variance is almost 4.5, 5 times less in treated compared to control. But that means the samples are stable in terms of aflatoxin content in various lots. Okay. So this was for maize, and this is for groundnut in Senegal. Again, these are huge big lots, and you can see that the level of contamination is in the treated is less than 4, and in many cases it's very high. And again, the variance has gone down quite dramatically. Okay. So this is quite an effective product. Now, why does this happen? As I mentioned earlier, there's displacement. We actually track these strains, the active ingredients in the soil and in the grain before and after application. So before application in the soil, there were low levels of the active ingredients, and the reason is because they are actually native strains. They're already present in nature. But in the grain, in the control, there was hardly any increase. But in the treated, about 70% of the isolates that we recovered from the grains were actually same active ingredient as the product. And the same kind of result was also seen in 2010. So in both cases, and this is in case of maize and in case of groundnut, similar trends were actually visible. And so you do get a displacement of the toxigenic strains with the non-toxigenic strains in the treated plots. Not only that, but we do see a carryover of the inoculum, which gradually goes down. But 1 year after treatment, 71% of the isolates in the field, in the field soil, were actually the treated, the active ingredients in the treated product, the product that was treated in the previous year, and it went down to 28% in 3 years. So we do actually need to reapply every year for better control. Now, if we have biocontrol and if we also have resistance, we don't actually, unfortunately, we don't have resistant maize varieties or hybrids that are commercially available. I mean, most of the lines that have been developed are lower yielders, and the farmers do not normally produce them. But there have been some very encouraging developments recently, and we hope that in the next few years we will have high yielding varieties and hybrids released in the African agriculture system. But what I want to show here is what happens if you combine biocontrol and resistance. So these are 5 varieties, experimental varieties. 3 of them are resistant and 1 is susceptible. If you do not apply biocontrol, these are the aflatoxin levels that you see normally, but if you actually convert them into percentage, if you do not apply biocontrol, you do get 66 to 88% reduction compared to susceptible, just with resistance alone. If you use biocontrol on these varieties, irrespective of varieties, the resistance, the control is in between 74% and 92%. And so it doesn't matter what the variety is, you do get a reduction. Okay? If you look at the impact of biocontrol and aflatoxin, and biocontrol and resistance is about 97% reduction, which means that if you can combine biocontrol and resistance, we do get very high levels of reduction that can reduce the aflatoxin level to much less than 4 parts per billion, which is very promising actually. Now, until now, we have in Africa, we have registered 14 biocontrol products with the national regulatory agencies. And these are registered in 10 countries. And there are 3 other biocontrol products that are registered in the world. One in the US, this is the first one that was registered, AF36. Aflagar was also registered in the US, and AFX1 was registered in Italy by Pioneer. Totally, I think we have registered more than 64 active ingredients, and that's quite a bit actually. Various scientific committees within the regulatory agencies, they have considered the products to be safe, effective, and of value to farmers. And these are just 2 examples of PCBB, which is quite a good regulator in Kenya, and the Environmental Protection Agency in Ghana. Now, after registering the product, we didn't stop there. And normally, in normal circumstances in an advanced country, these products would be given to, after development of the product, not registering the product. After development of the product, they'll be given to the private sector. But unfortunately, in Africa, the private sector is not that well developed. So we took on the actually task of registering the products ourselves. And that was quite a bit of an effort because we had to prepare various kinds of dossiers, do toxicological study, ecotoxicological study, and based on that, the registration was actually provided. So after innovation and development of the product and registering the product, we then went ahead and assessed market to develop strategy. We then selected partners for manufacturing and distribution and transferred the technology to them and licensed the technology to them and helped them to produce, market and distribute the product. And from this exercise, we learned, we adapted ourselves that helped in further scaling. And to do this, we must realize that this is not the job of plant pathologists alone. So we had a team of plant pathologists, engineers, social scientists, communication specialists, business development specialists, which is really important here. In addition to that, private sector investors, administrators, and donors helped us to a large extent. But the investments in terms of manufacturing and distributions was of the private sector itself. Now, we are working in 21 countries, and if you actually look at this, we have a pipeline that we follow, a process that we follow from finding out what kind of problem is there in each country to working with investors to scale up. And in addition to that, we have finding the native toxigenic, the atoxigenic strains, the active ingredients, enhancing the capacity, registration, commercialization options, the private sector mobilization, testing the market, and so on and so forth. So in various countries, depending on the time when we started the projects, we are at a various different stage. I'll show you a snapshot of a map later on that will give you more details than this. We implement an aflatoxin management system, which is centric on which is based on biocontrol, but just biocontrol is not enough. And so what we have is a system which is grounded to policy and advocacy support from the government, from the public sectors. And the public sector also helps in awareness creation and sensitization. The private sector is also important. The FPOs, what we call as FPOs, we call them implementers in Africa. So the farmer producers organizations and the farmers group, the value chain, we try to organize the farmers or help the implementers to organize the farmers into groups. And these FPOs provides inputs and services to these farmers. And at the end, they would actually aggregate those, they would aggregate the grain and use post-harvest management. And then we help them to create market linkages. And one of the input here is AflaSafe. But in addition to that, there are also yield enhancing and agronomic practices that are trained to the farmers, that are provided to the farmers. The public and the private sector, they work together on training to improve productivity, training for aflatoxin management, and aflatoxin testing. And based on this, with the market linkage, these aggregators then can actually get an incentive from the market, part of which is passed on to the farmers. So we actually did this in Nigeria quite successfully, and it is now being scaled up further. So if you look at AflaSafe at a glance right now, this research is continuing in 20 countries. As I mentioned earlier, 14 products are registered covering 10 countries. $6.5 million have been invested by the private sector partners in manufacturing and distributions. There are 4 factories operational, 3 are under construction. The 4 factories that are under operational are in Senegal, Kenya, Tanzania, and in Nigeria. And in Nigeria, one of the investors, they have built one of the largest plants for biocontrol in the whole world, actually. They invested a large amount of money. And I think this year they produced about 1,900 tons of AflaSafe. 3 factories are under construction in Sudan, in DR Congo, and in Mozambique. There are 10 companies that are manufacturing and distributing in 15 countries, and there are more than 100 distribution points, which will increase further. Until now, about 5,100 tons of AflaSafe have been produced to protect about 500,000 hectares. And using this, more than 1 million tons of AflaSafe, aflatoxin safe maize and groundnut have been produced. And this is where the million actually comes in the title. The countries that are in green are the countries where the product is already registered. And the countries in pink are where the testing is going on right now. And the countries in yellow are the countries where the strain development work is going on. So these are the 4 pictures of the 4 countries where we already have manufacturing plants. And incidentally, these are not, these are owned by the private sector. And except for this one, the one in Calro, which is owned by the government manufactures it, and they have a distribution partner, which is for the private sector corporate. Some impacts here. The aflatoxin management system that I mentioned earlier, what we had demonstrated, and this is actually done by an external agency, they demonstrated that that particular project increased the net income by 16% per smallholder farmer. The consumption of maize, safe maize, went up by 13%. We have also seen revival of groundnut export in Gambia and Nigeria. In one irrigation scheme of the government, they used AflaSafe in that scheme, and they could produce enough grain to feed about 500,000 Kenyans in drought-prone area. As I mentioned earlier, AflaSafe has been incorporated in the National Agriculture Investment Plan in 6 countries, and the private sector investment of $6.5 million have taken place. That helped in job creation as well. However, there's a lot more to done. The uptake is still limited in aflatoxin concentrate's core markets, which are dominated by large-scale producers, exporters, and the government procurement. And we need to actually now penetrate gradually the smallholder farmers who can buy the product themselves and use it in their farm for improving their food safety. Aflatoxin management, we had published a paper in Global Food Security, and we demonstrated that 15 of the 17 SDGs can be influenced by AflaSafe-centric integrated aflatoxin management system. I would encourage you to actually read the paper. If you want, I can send you. This is an open access paper, by the way. So what are the actions that are needed to scale up aflatoxin? So I mentioned about a few things. I don't know how much time I have. Can somebody mention how much time I have? My clock is not working.
No, please carry on, sir. No.
Okay. Now, we are also trying to improve the various production, the manufacturing process of the product, and also helping the manufacturer, distributors, aggregators. So in terms of improving their operations. So here is one example where we are developing new systems to improve the manufacturing. So normally, if we're producing the active ingredient, we need an advanced lab, clean lab, well-trained people. But oftentimes, these labs are expensive to actually build and not easy to handle. So my colleague Alejandro, he developed a system for producing dry spores in a central lab in Nigeria, and then shipping these dry spores to the manufacturers. And the manufacturers then just use the dry spore to create the active ingredient suspension for coating the seeds, for coating the grains, the carrier with the active ingredient. And this has actually helped quite a bit in terms of efficiency. About 2,000 tons of AflaSafe have been produced. We produced about 40 kg dry spores. And the production took place in Nigeria, Senegal, Tanzania, and Sudan. And about more than 200,000 hectares have been treated using this particular system in several countries. Now, until now, we have been doing this manually, but now we are automating the whole system so that we can produce much more dry spores in tens of kgs in each batch, so that for manufacturing and distribution partners, they are serviced and their operations actually improve. Incidentally, the manufacturers, they actually purchase these dry spores. They are not given to them for free. Another cool intervention that has actually taken place is about in situ testing. So normally, for farmers and aggregators to know what is the level of aflatoxin in the crop, they have to often sample the crop properly and then send it to a central lab. And oftentimes it takes many days and sometimes weeks to get the results back. And if the aggregator or the farmer is sitting on this grain and waiting for the results, aflatoxin keeps on increasing. And so they need to know quickly how much aflatoxin they have in their grain lots. So what we did was to actually adapt a lateral flow device method to take it from the lab to the field. And so the analysis is now done in the field in front of the farmers, and they can actually see it being done, and they can see the results because you get a digital display of the amount of aflatoxin. And they can see the results themselves and be convinced on and know what kind of aflatoxin they have in their crop. This also helps in the farmers and processor in trusting various technologies that are offered for aflatoxin control. We are now trying to actually develop a system by which we can use this. We can actually put it in a box and take it behind a motorcycle in the rural parts and the markets of Africa. FAO has a similar system called Blue Box, where they do many quality control, quality tests. So we're adapting that to see whether we can actually get it done. We can put it behind a truck, behind a motorcycle, and get the testing done right in the field itself. Now, there are many challenges of technology transfer. So once you have the technology, that's not the end of it. And technology transfer is not an easy thing. There are many challenges in technology transfer. So the ideal scenario in our case is that we register the product, which itself is not easy. We develop commercialization strategies. We find manufacturing and distributors. We have farmers demanding the product because we do the testing along with them and the industries are committed to pay a premium for prices for the safe crop. But that's not what happens in real life. Okay. There are some factors that can actually affect implementation: is political stability, conflicts. Sometimes the infrastructure in terms of moving material is not adequate. The crop and market trends and availability of market incentive is an issue sometimes. Timely delivery of materials for manufacturing is sometimes a problem because some of these chemicals are actually transported from the US. There are availability and proper and training of manufacturers, that's also sometimes an issue. Oftentimes, the order for production comes very late, and so the timely production, distribution, and quality control becomes an issue sometimes. And training of farmers have been real, it's not a challenge, but it's a must in order for the technology to be correctly applied so that they get good control. Plus, of course, the COVID-19 disruptions have taken place. So some final thoughts. So the key implementation messages are: we need to create awareness about aflatoxin and its management, which is low right now. Use of biocontrol as a key component of aflatoxin management system, as I had mentioned earlier, described it. Creation and promotion of testing facilities, because the users of technologies, they must know that they have proper control, and they only can actually determine whether they have proper control or not by testing. It's just like COVID. I mean, unless you actually test, you don't know whether you have COVID or no COVID. The premium for aflatoxin from aflatoxin conscious market to startup, that's an important aspect. I can't overemphasize the importance of public sector support, such as input supply scheme, as well as the regulatory support that are required from government institutions, particularly in terms of monitoring and implementation of standards in the marketplace. Overall, it's not just technologies that are actually important. We need to converge technical policies and institutional innovations to deal with the problem at the ground level. Neither of these three by itself can handle the issue, they need to be combined. And this is a lesson that we are learning in many places actually. And this is the area that we are actually emphasizing right now in our implementation strategies. We have been working with many partners, as I mentioned earlier. These are some of the names of the organizations, both private and public sector, with whom we are working. Like we've got many research organizations in many African countries. In addition to that, we have industries like Samil in Sudan, Bamtare in Senegal, Harvestfield in Nigeria, Copert in Kenya, Macrofertile and Diageo in Ghana, Stafito in Burkina Faso, A to Z in Tanzania, UPL in Mali and Ghana. So there are many private sector organizations with whom actually we work, in addition to various donors and national institutions. We are fortunate to actually have support from various donors, including the Gates Foundation, USAID, the Australian government, the UK government, and many other organizations. We have a team of people with diverse functions, both for DNA technologies, which are most of the people are primarily based in USDA ARS lab, because this effort that we have done is in partnership with USDA ARS, with my colleague Peter Coty and in ARS. In addition to that, we have plant pathologists who have been involved in product development and field testing. We have the support from various important people who have advocated for our project from various organizations. Commercialization and business development staff, they are key to finding the investors and then supporting them. We have engineers who have actually worked on the design of the manufacturing plants. So all the manufacturing plants are actually designed, and they help these investors to set up the manufacturing plant. We provide the technical support in terms of designing and erecting the manufacturing plant. And we also have people from socioeconomics policy, and we have breeders. And we have support from many graduate students who have actually worked at product development. Thanks for your attention, and I will welcome any questions or comments.
Thank you, sir, for your excellent and very knowledgeable lecture on biocontrol-centric aflatoxin mitigation in Africa. Now the questions are invited from the participants.
Sir, kindly unshare your slides, sir.
Okay.
Yes, Dr. Ravi Katrapal, sir, please.
Thank you, Jambul. Thank you so much, Ranjit. It was music to the ear, listening to your contributions from ground zero to something going regional and global. I think it's a very huge contribution to science, farmers, and you develop all the appreciations from IPS and from every one of us for your patience to stick on. It seems you had married, aflatoxins. You stick on. You didn't only have a partnership, but you really married this field of research, which really paid you in the long run. And a good marriage pays always. Well, with that background, I've seen the way you have done it, especially in Africa and with support from US or USDA. I'm a bit selfish because I work for Asia and the Pacific. So I'll come to my point, try to distract you a bit. If we are doing some SKDF WTO projects here, where our basic aim is facilitation of trade and compliance to either Codex or the IPPC or OIE norms. Your topic, actually, I had briefly probably hinted you once, to talk on developing a kind of SPS capacity related to aflatoxin detection, mitigation. In mitigation, I mean all methodology you have shown, especially I like the storage thing, which really comes very handy and doable in the development world. And plus, there are a lot of things which are related to lab. And if we have something like AflaSafe available, but still we really need to look at more and more non-toxic aflatoxin strains, which I think will be key. So from the Asia level, I do not know where we are, number one. You might be knowing. We need a very good gap analysis. I know Dr. Suslinder Desai has worked a lot before, and we had been banking upon his expertise in India to respond to any WTO queries once upon a time. But Asia, where we are, and with that knowledge, can we build an SPS capacity building for safe trade by mitigation of aflatoxin related issues, complying to the codex issues? And many times there are both sanitary and phytosanitary issues. It is not only what you talk is a sanitary issue, aflatoxin. And there is a big problem if you talk of SPS value chain. When you talk of value chain, a poor farmer has to deal with both. I have seen it with ginger in Nepal. You know, if we can have a holistic capacity building where we have, because sometimes these sanitary issues become a phytosanitary issue, or phytosanitary issue becomes a sanitary issue, like you have more aflatoxins in storage and all. The question I want to pose is, do you have a bit of gap analysis for Asian countries? I'm more focused on South Asia and Southeast Asia, in fact. And now we have started working in West Asia and maybe a bit in the Pacific. Pacific, we don't have much knowledge. So if we have some good information, we can package it into a beautiful project with your expertise. And we can think of collaborating with some of the countries within Asia who can help some of the countries who need help. You know, we can bring two kinds of partners from STDF project. And we can really move ahead to resolve this issue of aflatoxin, which I think is a great burning problem with your initial PowerPoints, what I saw. And I should thank you for acknowledging each and every, even the smallest denominator who contributed in last 20 years in the whole exercise. That was the most powerful PowerPoint I could see, the last one, your team. Thank you. Over to you, Ranjit.
Thanks, Ravi. You know, we did exchange some, I think we need to actually firm up the discussions offline maybe. I agree with you. And, you know, the last 22 years I have been with IITA, the attention was Africa because IIT's effort is Africa-centric. But we do have an interest, and we are gradually expanding that interest. And we do get actually inquiries from Southeast Asian countries about biocontrol, for example. Bangladesh, I think, is going to start a project soon. So that's one. Most likely, I've not actually heard the final end of it, but Bangladesh might actually start a project soon. But we would actually welcome working in Asia, because, you know, as you know that with one CGIAR, our effort is no longer confined to Africa. We work now in the entire world, in the entire developing world. So yes, we can actually we'll welcome this opportunity to work with you to develop a project. Incidentally, we actually helped the Nigerian government to develop a project on sesame and shea nut control for aflatoxin, and which included training, and it included capacity building, then infrastructure in terms of drying, storage, and all that. So that was an STDF project that were concluded about 5 or 6 years ago. Right now, we are working with a private sector organization, an STDF project that we helped a private sector organization in Burkina Faso to get again on aflatoxin control and maize. Okay. And there again, it's the entire value chain aspect that has been actually taken into consideration. Right from, they're actually helping in setting up new manufacturing processes, the training farmers, the traceability system, and it's a whole entire value chain being actually being addressed. The third project that we are working on right now is again in Burkina Faso with another trader, but this is not through STDF, but West Africa Trade Hub. And again, it's trade related. Okay. And so there are these opportunities, the experiences that we have with which we can actually work with in Southeast Asia as part of the plant health initiatives. You're aware of that.
Yeah, part of it rather, yeah.
We will welcome that.
Sorry? Yeah, go. Thank you. I think that's a very positive response and we can carry discussion because I would like to leave time for others to ask questions. Right. But thank you so much. I really enjoyed your presentation and we'll look forward because one of my colleagues here, Dr. Sasi Rekha Rajendran, she is also there from, she just did her PhD in aflatoxins. She's joined APARI. So with her talking of developing a project for SPDF. We already have one going on, one more we just submitted. So we would really like to take anything which is related to trade and economy linking with science. Yeah. Thank you.
Dr. Desai, please.
Yeah, thank you. Thank you, Dr. Jambulkar. Good morning, Ranjit. How are you doing?
Good morning, Desai.
Nice to see you after a long time. Thank you, Ravi, for making that mention of me. Thank you very much. In fact, that was one of the best things what we had. At this point of time, I just recollect our late Dr. Nagarajan, because Dr. Ravi and Dr. Nagarajan, they were there in Kasargod when I was presenting my paper on hazard analysis, critical control point analysis of aflatoxin contamination management, because we identified that there are about 7 stages in peanuts which get affected because of the aflatoxins. And so we addressed each point by giving due weightage to that with which we could successfully manage the contamination to tolerance levels. So I remembered those days now once again. Thank you very much. A couple of observations. One good point is that only about 40% of the Aspergillus strains are aflatoxigenic. That's in fact a blessing for us. If we are going to hunt for them, I think we are easy to get quite a few of them. And I also recollected Peter Cotty's work. You also mentioned about it. And then in NPRL Dawson, they registered Aflagard long time back. The second thing is that we also thought of these aflatoxigenic genes, but then non-aflatoxigenic strains. But then we thought that in our study, we identified that 1 out of every 5 non-aflatoxigenic strains are cyclopyogenic acid producers, which is equally potent problem. So then we resorted to trichoderma, which is a safe bet for us, because in Saurashtra, for me, in groundnut, sclerotium is also a major concern, which takes about 30% toll of the plant. So this trichoderma can manage me both Aspergillus as well as sclerotium. So that's why we banked upon that. Now today in Saurashtra region, trichoderma is a common application system in groundnut production systems. And then finally, 2 things I just would like to ask you. The market linkages and all those things, Ravi was also mentioning about it. Actually, the loophole in this is that I send my product to EU after my certification from Mumbai, and then SPS, SPG will do certify, but then at the importing port, if it is rejected, the trader, the vendor doesn't bring it back, but he will divert it to Southeast Asia, where limits are little bit higher. So that's an easy way for them to escape, in fact, so that people are not very much serious about managing the contamination problem. Then lastly, my question to you is that, what is the shelf life of your this Afla safe what you produce? Thank you.
Thanks for the questions. And I know that you have been working on aflatoxin for a very long time, since I think the time when I was at
This is past time now for me, aflatoxins. That's all. Okay. Now I am into teaching agriculture. Okay.
Now, in terms of the biocontrol part, the strains that we actually select are both non-aflatoxigenic and doesn't produce CPA. And even if it does produce CPA, it's very low quantity of CPA. If it does, okay.
That's great.
So that's the reason why the selection process is so important. It's not just finding an aflatoxigenic strain, but not only the non-toxin producing capacity for aflatoxin and CPA, but also their genetic group. They must be widely distributed.
Perfect.
For them to be adapted. That's a very crucial aspect, because you're going to use these products across a country which has got many agricultural zones. And so they must be widely adapted. And one of the reasons why we use a multi-strain product is because it's wide adaptability. Okay.
Excellent.
Now the other part, yeah. Now the thing about the trade aspect that you actually mentioned, okay, this is very common practice. I mean, if you take the example of US, the US, they would actually, the farmers, they will take their truck to the elevators, and if one elevator rejects it, they'll go to the second elevator. And if none of the elevators take it, they will drive it into Mexico. You know? Exactly. And so in this case, the trader would actually lose money by diverting the product into Southeast Asia from Europe, because he or she is not going to get the premium prices of Europeans compared to Southeast Asia. So it's going to be a loss. There is no doubt. It's not a complete loss. He's going to recover some money, but it's going to be a loss because the amount of investment that he's making to ship it to Europe and then diverting it into Southeast Asia, he's going to lose money, but not a complete loss. Okay.
One observation, I tell you, I was reading Maxwell's paper in 2021. What they suggest is that, is it possible that you mentioned when there is 6.1 level of aflatoxins, your atoxigenic strains were bringing it down to 1.7, and then when it is much higher, then it is bringing down to by 93%, when it is 53% toxin levels. Is it possible that these atoxigenic strains are also detoxifying the aflatoxin as reported by Maxwell?
Yes, it does. In fact, there is a paper that just came out 2 months ago.
Yeah.
The first author is Lorena. Okay. It came out in Plant Disease. Just check the last 2 issues.
Yeah. I read Maxwell's paper on this. Okay, please go ahead.
So these strains are actually known, the atoxigenic strains are known to detoxify aflatoxin, because they actually grew the toxin-producing aspergillus with aflatoxin in the medium. Okay, and these fungi could actually grow with aflatoxin as the sole source of food. Okay, and they could degrade it. So degradation is
a carbon.
But the major effect is actually on displacement. So there are other factors that are also available. Okay, there's something called touch inhibition. So toxin producer and non-toxin producing, they touch each other, and then detoxification takes place. But the major effect is due to competitive displacement.
Thank you. I recollected 1986 and 87 when we were discussing outside 301 building. Thank you. Yeah, please.
This is Dr. Sridhar from Chennai. It's one of the best lectures which I ever heard. And as of now, since we have the biocontrol hand, much developments is going towards how to prevent the contamination in human and animal food chain contaminations. And there is a lot of private and public partnership that's going on. My hearty congratulations on the project. But my interest centers around the ability of plants. If I remember, I think that there are some correlations between the insect resistance of the plant as well as the drought, the abiotic drought, the environmental conditions. That has got some relationship with the aflatoxin production, reducing the aflatoxin production of the plant. Because if we can capture a point where we can reduce the plant's ability to produce aflatoxin, probably we may have a better option for managing this. It's just an imagination. And with the advancement of genomics and studies related to genome-wide accession studies, locating the genes and all these things, I think your coordinators, the U.S. Department of Agriculture Research, lab facilities may already be thinking about it. If we can locate certain genetic mechanism that can be manipulated now very easily with all these techniques, in spite of the policies related to government policies related to against the genetically modified organisms and that should not halt us from approaching this area. Yes, probably I think in future it may be possible to come out, because in the absence of resistance in the maize genotypes itself for aflatoxin production, it's also possible to weight the genomes in such a way that we can try to trace the aflatoxin production, the entire genetic mechanism, and then manipulate the other way around indirectly to reduce the aflatoxin production. I know I'm not seeing much papers on that, but except a 2015 paper on PLOS on effect of drought resistance, that because the drought resistance itself is a very complex phenomenon to work genetically. Probably, I think there are some opportunities if some people are thinking about these and try to pursue it. Your comments, please.
Thank you, Dr. Raghunahan. You were right, actually. Insect resistance and drought resistance, it does have an influence. And so when we actually talk about adapted varieties, we talk about drought tolerant varieties and insect resistant varieties, because insects actually provide the avenue for the fungus to get in. In terms of, there has been a lot of work done on the genomics and transcriptomics of impact of drought on aflatoxin. USDA ARS, they have done quite a bit of that work, and including there's a group in Mississippi, Marlene Warburton, who has also actually looked at this. A large amount of effort has gone into this in the US, but unfortunately, and one of the major professors in Cornell, I was talking to her, Rebecca Nelson, and she has been working on this for some time. And what she says is that when they begin to incorporate all these traits into maize, they get the resistance, but somehow the yield drops. And so there have been a challenge in terms of combining both the resistance and the yield. Hopefully that challenge is going to be resolved soon, because with time, people will find various ways of actually dealing with it. In terms of resistance to genetic modifications, there have been quite a bit of paper of late. I mean, there's one from Arizona State University, and they have used Higgs, host-induced gene silencing, and that was quite effective. And ICRISAT has been doing some work on this, and I think about a year ago, there was a paper from ICRISAT that looked at two ways of actually silencing, reducing. One is silencing the gene for producing aflatoxin, and also reducing the Aspergillus population, Aspergillus biomass. And I think most recently, about 2 months ago, there was another paper from, I think, from Cheng, and they use a different set of genes, again, through Higgs approach, and that was quite effective too. But the major issue with that is, when will we get regulatory approval to get to use?
In fact, I was associated with Dr. Rebecca Nelson introducing the marker-assisted technology to India from the Philippines. Really, these challenges remain. Probably, I think that this is one way of approaching at least a small fraction of labs somewhere in the country, in India.
Just to supplement to what Ranajit said just now, we had experienced that the wireworm damage in peanuts predisposes the peanuts to aflatoxin contamination. It is a very common feature, and especially water activity, as you said, is one of the most predominant parameters which predisposes. Immature pods especially, they get very quickly damaged and then it happens. Yes.
That has to be pursued.
So they say there's a series of paper by Naresh Magan from Cranfield University, who actually looked at the impact of water activity, temperature, carbon dioxide on Aspergillus growth. And initially they started just the physical effect, but later on they dug into more into the genomics aspect of it.
Exactly. Yeah.
Thank you.
Yes.
Are there any questions in the chat?
Yes, sir. One question in chat box, sir. Application in open field condition, there is a possibility strain mutant atoxigenic to toxigenic.
Right. So this has been a common concern, and we have heard that many times. Now, again, the way that we actually select the strains, and we put a lot of effort in strain selection. So all these strains that we actually use as active ingredients, these strains have defects, many defects in aflatoxin biosynthetic pathway, not just one. There are many defects. In some cases, the entire biosynthetic pathway of 26 kb is missing. Okay. And so that's one. Second is, it's not just the atoxigenic strains that we actually use. We actually use a genetic group. The active ingredient belongs to a genetic group that do not contain any toxigenic strains. So they are completely atoxigenic vegetative compatibility groups. Okay. And so the chances of mutation occurring and becoming toxin producers is very, very slim. And even then, if it happens, it is already happening in nature because these strains are natural strains. So we are not actually introducing something that is more risky. If the risk is there, it is already there. But the way we are actually selecting the strains is such that the risk of those textual recombination or mutation is reduced dramatically. So the strain selection is actually crucial in how you develop products. I have seen some papers come out, they would actually screen 100, 200 strains and get 1 or 2 atoxigenics and then begin to use it. But that actually poses some risk as well. The strain selection has to be really thorough.
Yeah, but does it also hold good for, because it's definitely an intriguing question as to why does the Aspergillus take the 20 steps in producing ultimately that aflatoxin? What does it get out of it? That's what's an intriguing question for me always. But the metabolic pathway and all what we are searching for, does it hold good for CPA and others also?
I didn't get
cytopyrazinic acid and other toxins.
I didn't get you. CPA for what?
When we are searching for this, the VCG groups, which are non-aflatoxigenic, does it also hold good for the CPA or other toxins also?
So The group that we actually use, these are atoxigenic VCGs for aflatoxin. You know, the CPA, there was a study done, it is not published, but it is a study done. I shouldn't say published. I have actually looked at the literature on CPA, and I found that the maximum amount of CPA that has been found in any crop is much lower than what people think are the toxic levels. Okay. So I mean, compared to aflatoxin, CPA's importance is way down to the extent that CPA is not even regulated. Aflatoxin is regulated. Okay. And I mean, if you actually take that instance, you don't even know what resistant varieties actually have. Nobody looks at the risk assessment of resistant varieties. I mean, what kind of alkaloids, for example, would some of the resistant varieties produce? Okay. So in terms of risk, aflatoxin has got a much bigger risk than CPA.
I just want to take this platform to tell Ravi that I can also be an asset for you in this regard if you want to go together. Thank you.
Can I add a point? CPA is almost like a virulence factor of the pathogen. It aids in creating a this conducive atmosphere and creating a disease in the plant tissue that is there. So the formation of CPA is very simple, not as complicated as aflatoxin. But it has got more role in producing the disease in the plants.
Exactly.
Any other questions?
Malgam Singh,
Yes, sir.
actually, my point is we have very few people in India working on aflatoxins.
Yes,
I
sir.
think
Yes, sir.
first IPS should focus on that as to, because when I was working, my look was only at Bihar, that one laboratory which was working at that time. And afterwards, today very few people are there. I think
Yes,
that's
sir.
where, first of all, we need to build up the research capabilities of the people. Thank you.
Then yes.
I completely agree with this. Dr. Thakur, who may be in the audience,
Yes.
Dr. Thakur and I, we did a review of aflatoxin situation in India. I think about 4 years ago. There was an IFPRI project, and we actually got the same situation. And when I look at there was a lot of work done in India earlier, about 15, 20 years ago, and including there was a all India coordinated project on mycotoxins. And a lot of good information was actually generated. But in terms of, and the groundnut group, for example, if you look at Dr. Basu's work, Desai's work, Ikrisat's work, a lot of good work was generated. But I think in the Indian system now, I don't see that much of an emphasis on aflatoxin research. Not only aflatoxin research, I would actually like to see application of the research, because that's where most of the impact is. It's not just the research for research's sake, but application of it to reduce the burden on the industry and the farmers. That's where, as a plant pathologist, we can make greater impact in addition to, of course, generating knowledge.
Exactly. Because I was looking at Bhagalpur. After that, Bhagalpur lab closed. And now Ikrisat and Junagadh, these are the two centers which are working. And nowhere else we find people. I don't know. Either facilities are not created or people are not. We don't know what exactly the reason is.
I know that the industry is actually working on it.
They're interested.
Yeah, the industry is interested.
Yeah.
And there's a lot of effort going into the genomics side of it gradually. But I think we need to actually work on it at the grassroots level and solve the problem there. And particularly with the increase in maize area, and compared to sorghum, I mean, sorghum and millet are being displaced by maize. And as maize is being taken to more wetter areas, Bihar, in other places, the contamination is also increasing. When I was in Nepal 2 years ago, and the Nepalese were saying much of the contamination that they get oftentimes are the imports from India, from Bihar and other places.
Yeah.
And the similar situation is in Bangladesh. Bangladesh, it was a rice producing nation. And look at the amount of expansion of maize into that area, in that wet area. And that creates a lot more problem actually.
Yeah.
Thank you, sir.
Ravi, just
Any
now I
other,
was telling,
any other more
just now I was
questions?
telling Ravi that I can be part of your program also. I can extend my support. You were not there. Thank you.
Thank you.
Dr. Bandyopadhyay,
Yes, sir.
I am, I am virologist still. I enjoyed a lot your
Thank you.
topics. And I have seen you after a long time, and Khetropal sir also. I have seen you a long time. And I am very much happy to our President, Madam, and Dr. Gogoi. They have created such kinds of lecture series of Platinum Jubilee or IPS. In this time also, we are seeing many. And today is the working day. That's why audience is to some extent anyhow, because the good lecture. I'm very much happy. Khetropal sir, how are you?
I'm fine, thank you, Kajal. How are you?
And Siddhar sir. Smiling face after a long
Thank
time.
you.
Siddhar sir, smiling face.
Thank you very much.
I now live in Delhi, if you don't know. Huh?
Yes, yes. Oh, you are living now?
Yeah, I live in Delhi.
Okay, sir. Okay.
I think
Okay.
no more questions, sir. We come to end this lecture.
Thank you, sir. Now I request Dr. Malkan to give vote of thanks.
So, Namaskar. On behalf of society, I feel immense pleasure to propose a vote of thanks. I am Dr. Malkan Singh Gurjar, a treasurer, Indian Phytopharmaceutical Society. The society is celebrating 75 years of journey. They made a tremendous contribution in particular Indian phytopathology research in India and abroad also. At the outset, I extend my profuse vote of thanks to Dr. Ranajit Bandyopadhyay, principal scientist and leader of Africa-wide Alpha Safe Initiative, IITA Ibadan, for accepting our invitation and deliver the 24th 75 lecture series celebration of Indian Health Society. He graced the occasion and delivered this lecture on more than 1 million reached a 2-decade endeavor of biocontrol-centric aflatoxin mitigation, in particular in Africa. He emphasized health standard of aflatoxins and food safety standard and biocontrol, particularly alpha safe, how it is utilized in field, how many countries it is utilizing, and what is the impact of this particular biocontrol agent to manage aflatoxin. I would like to thank our President, Dr. Pratibha Sharma, and President-elect, Dr. Rakesh Pandey, and Secretary, Dr. Robin Gogoi, and Joint Secretary, and all the executive members of society, and editorial board also, the senior editor and chief editor also. And also I would like to thank Ravi Khetropal, former president of IPS. Sir, very much thankful to you joining for this lecture series. And my special thanks to all the participants and all the IPS members who joined today's lecture, and it makes a grand success. And as rightly pointed out, this topic is, you can say, very less people are working on aflatoxin. So we try to do more work on particular aflatoxin. Thank you, one and all.
Have a good day.
Thank you.
Nice
Thank
to
you very
see
much.
Thank you, sir. Thank you.
Yeah.
Thank you very much.
Thank you.
Dr. Desai, thank you very
Thank you,
much.
sir.
Thank you, Kajal.
Yeah.
Okay.
And Kajal,
Bye.
Sir,
Bye.
I missed your lecture because of some overlaps. In fact, I was very keen to attend your lecture.
no, sir, it was postponed because that day was a
I
Saraswati
see.
Puja.
Very good. I am so happy because I am missing it.
The cotton leaf curl.
Please keep us in